Welcome to The Pulse, a podcast that dives deep into biotech stories and features biotech leaders. I'm one of your co-hosts, Fang Xie.
I'm your other co-host, Yosef Baidi.
The podcast is brought to you by the Harvard Biotech Club, a students-led, Harvard-affiliated biotech organization, which is on a mission to bridge the gap between academia and industry.
Today's guest is Dr. Bill Sellers. Bill is a core institute member of the Broad Institute of MIT and Harvard and serves as director of the Institute's cancer program. He is also a professor of medicine at the Dana-Farber Cancer Institute and Harvard Medical School. Bill spent his academic career at the intersection of cancer biology and using genomics to investigate mechanisms of tumor development. Of note, he collaborated with Matthew Myerson to initiate the Broad's first major cancer genomic efforts, leading to the discovery of EGFR mutations in lung cancer. Previously, Bill had served as VP/Global Head of Oncology at the Novartis Institutes of Biomedical Research, where he oversaw small molecule, antibody-based, and cell therapy drug discovery efforts in oncology. In addition, he initiated and led the Cancer Cell Line Encyclopedia Project, which was a collaboration with the Broad Institute. And most recently, before starting his academic lab the first time, he had completed postdoctoral training in the labs of Dr. David Livingston and Bill Kalin. So with that, we'd like to welcome Bill to the podcast.
Thanks for having me.
Great. Yeah, we're glad to have you. So we usually like to learn a little bit more about our guests before. So could you tell us a little bit about your upbringing, like where you're from, and a little bit about your family as well?
Yeah, so I'm actually raised in Massachusetts, which is fairly unusual for someone who's working in Massachusetts in the biomedical area. I grew up in Reading, Mass. My mom and dad met at Tufts University. My mom's from India. She's actually from Punjab originally and then from New Delhi after the partition. And she came to college at Tufts when she was 16 or 17. Met my dad, who's Irish Catholic. And so I'm from a mixed ethnic background. Grew up as I said, in Reading. Went to high school at Phillips Academy in Andover, then Georgetown University. Then came back to Massachusetts to the University of Massachusetts Medical School to do my medical training. And then off to the University of California, San Francisco for internal medicine, and back to Boston to the Dana-Farber, and been here ever since.
Cool. Yeah. So I guess circular, started in Boston, came back. And then you mentioned a little bit about your background. Are you like half Indian, half Irish Catholic? Do you kind of are in touch with like both traditions of your family? How would you say that was like growing up?
Well, certainly not as much on the Indian side. I've been to India 3 times, and I have my mother's twin sister also came to the U.S. and lived in Washington, so I have cousins from Washington. So certainly I know all my cousins in India, but yeah, clearly it was a little less influenced on the Indian side than on the more or less American side, let's say.
Okay. Gotcha. And then you mentioned a little bit about, your start of your academic career starting at Georgetown. So what, as a kid, what led you to become interested in science and specifically go to Georgetown to study biology, right?
Yeah, so in high school, I had 2 fantastic biology teachers, and this is going to date myself a little bit, but during, in between when I was a freshman and then I took more advanced biology courses, the model for how the plasma membrane was organized changed from the sort of this trilaminar structure to the fluid mosaic model. And also the chemoosmotic hypothesis for how the mitochondria generates energy also changed from this idea that electrons were literally transported to this the creation of the hydrogen ion gradient. And I was amazed that sort of some very basic aspects of cell biology were completely misunderstood, and that, like you could do experiments to figure out stuff that was sort of this essential part of life. So that was really the part I was attracted to. Why I ended up at Georgetown actually had very little to do with biology. In my junior year in high school, I spent a trimester on Capitol Hill as a Capitol intern in high school, working for William S. Moorhead, who was the representative from the 14th District of Pennsylvania. And so, I loved Washington, got to know Georgetown there, and went to Georgetown not really because it was great in biology, which turned out to be, it was pretty good in biology, but mainly because I thought the school was really cool and it was a great area to go. So I went to college as a history biology double major, and in my first history class I pretty much fell asleep and decided that I probably wasn't really that interested in history, and I focused on biology after that.
Interesting. History. Yeah. You were also interested in coding, back then.
Yeah, so in high school, I remember they got one of those like teleprompter things that you could program in BASIC, and it was this room, and it would like churn out this paper, and it was like deafeningly loud. So I learned how to do these basic little programs to make smiley faces and stuff print out on the computer. And then when I went to Georgetown, you had to take Fortran programming, but then I decided to take assembler language, COBOL. Database structures. So I did a lot of programming in college as sort of some side hobby, more or less.
Cool. Yeah. Imagine with biology, it's tough to keep up with that in coding, so you had to choose one to follow.
The coding was always more fun, actually. I know what the problem is people have. It gets very addicting to sort of fix your programming problems, and you spend hours in the computer labs. At that time, we didn't have laptops, so I loved that. It was a lot of fun.
Cool. And then, it sounds like you're very passionate about biology and love coding, and you're
history.
And the next step you had one on was go to med school. So, like, why did you get interested in medicine?
Well, as I said, I was really interested in the science side, and at the time, I would just say like the community of biology majors at Georgetown was predominantly pre-med. Mm-hmm. And so I would say I wasn't really even exposed to sort of this idea of getting a PhD. So it seemed sort of natural to progress to getting an MD. And so I wouldn't say it was any sort of super thoughtful thing on my part. It was just most of the people in my group were pre-med. I thought that sounded like a pretty good idea.
Yeah, I feel like it's very practical too. Like if you don't have scientists in your family, it's like if you like science, everyone knows what a doctor is, so you're going to go med school to do that.
Yeah, I mean, in retrospect, I don't think I really understood how diverse the opportunities were following an MD degree. It doesn't really restrict you very much. It actually opens up many, many more doors, but I wasn't thinking like that at the time.
Mm-hmm. Yeah, it's fair. And then, now we know you as like someone that's like so focused on research. So in med school, did you get a chance to do research while you were studying?
Yep. Not a lot. I actually, because of my computer background, I got a sort of part-time job with the stress reduction program. Oh. And they were teaching people how to meditate and sort of overcome pain and other things through self-awareness, and they were collecting all this questionnaire data from people and pain score data, and they had tons of data sitting around, and they had no one to sort of analyze it. So I put it all into like the SAS statistical program and then ran all the statistics for them. And then briefly, I had a short job trying to program a Mac to record blood pressure from a rabbit.
Oh, wow.
Yeah, it was very funny because it turned out the Mac couldn't keep up with like you would run out of data within like 30 seconds of recording a rabbit. So there was sort of some fundamental problems with the system we had.
Oh, wow. So, very different than what you do now.
Yeah. So what's your specialty back then? Yeah.
Well, so medical school, you don't have a specialty, but, your third year, you do all the rotations, medicine, surgery, and I decided internal medicine was the part I liked, and so, I applied for internal medicine residency and went to UCSF.
Cool. And then I heard from a previous talk you had mentioned a little bit about this, but at UCSF you had got involved with research as well. So how exactly did that happen? You find the time to do research while you're at this critical part of your training? Yeah,
so I think a lot of people know who Charles Sawyers is. He's a very prominent scientist. Well, Charles was my resident at UCSF. He was a year ahead of me, so he was a second-year resident, and I was an intern. And I think he was my resident for like 4 months of my internship, which is kind of weird that we kept ending up at the same spot. And he was very interested in research, and he figured out how in your third year of residency to take all your electives, put them together, trade them with somebody, and get 6 months of research time rather than doing the electives like cardiology, pulmonary, renal, that kind of thing. So I thought that sounded like a pretty cool idea. So I didn't quite get 6 months. I got 2 blocks of 3 months surrounded by December, and found a lab at the VA Medical Center that was working on trying to clone new translocations, and was working on what was thought to be an important oncogene then was called the PIM1 oncogene. It was identified as by insertional mutagenesis of a mouse virus. And so I started working on PIM1, and I was actually going to the clinic collecting blood from leukemic patients, consenting them, getting their blood, and then doing northern blots to look at PIM overexpression in leukemia.
Cool. So you're kind of involved with the whole step, getting the samples, analyzing it, and
that's
trying to use that insights to help with
it was really cool. Yeah, phycol purifying the blood cells, RNA extracting, running the northern blots. Yeah,
it's like the real physician scientist life. Yeah. And then, after your time at UCSF, you had done a clinical oncology fellowship at DFCI, so you came back to Boston, and there you had worked as a postdoc in David Livingston's and Bill Kalin's labs. So from that part of your career, what impact did this postdoc have? Now that you have your own lab in academia, was this like a pivotal moment for you?
Yeah, of course. Yeah, I mean, it meant everything, right? So during your clinical year at the Farber, you have to decide, do I want to be more clinical or more research? And we would have investigators come and talk to us about their labs. So, they would sort of pitch what their lab was doing. And David came, and it was like, I call it like the John Madden of science. It was like having John Madden. Yeah. Instead of explaining football, explaining, tumor suppressors and viral oncogenesis. And when David was through, I thought that they were going to solve the whole cancer problem, and they were going to do it so fast I wouldn't have any chance to work on anything, which turned out to be very naive. But this confluence of how viruses evolved proteins to bind and inactivate tumor suppressors, and then tumor suppressors like RB had just been cloned a few years before by Steve Friend and the Weinberg lab and Ted Dryja at MGH. That was like so amazing that the evolutionary conservation of the viruses evolved to tackle what turned out to be genetically altered tumor suppressors. Like sort of the beauty of that discovery was really amazing. And it felt like this is sort of what cancer is. Like this is how cancer works. And so, I was like super excited to join David's lab, and I joined, and at the time, Bill Kalin was a postdoc in David's lab, and he was working on this idea that the viral proteins produced by T antigen or E1A were working by binding to RB. They were binding to this region that the lab and Jim DiCaprio had described as the pocket, and that there must be pocket proteins that were being disrupted, and that disruption was the key to understanding why RB was a tumor suppressor. And so he developed this super cool way of trying to clone proteins bound to the RB pocket by making cDNA libraries, expressing RB as a radioactive protein, and then fishing out cDNAs by expression cloning. And he had cloned a whole bunch of these, one of which he called REBAP1, one of which he called REBAP2, and REBAP1 turned out to be E2F1. So when I joined the lab, they didn't know that.
E2F1.
They were trying to figure out what it did, and the first thing I did was to help clone the full-length E2F1. And then I ended up working on this other protein, RBAP2, which turned out to be very difficult, and I'd never really figured out what it did. But that was my introduction to science, and Bill obviously was very successful then and got his own lab, and so I'd been working very closely with Bill in David's lab, so I just moved down the hall with Bill's lab and eventually started working on this idea of the RB-E2F complex, and that it was more just, people thought at the time, RBE binds E2F and blocks its activity, and we thought that probably there was more to it, and that the RB-E2F complex itself was a repressor, and that there were really two things: RB bound to E2F as a transcriptional repressor complex, which suppressed below basal activation, the promoter, and then when you lost RB binding, E2F becomes free, and then it comes activated. And those were two different states that were different from the unoccupied promoter. So, like the mechanistic insight to exactly how the RB tumor suppressor function was, super exciting to me, and it's still exciting. In fact, I like that's the part I like. Like, you find out something's involved in cancer. Exactly what it is doing is important, ultimately, for figuring out if there's a therapeutic route forward.
Yeah, that's a good point. I feel like a lot of us that are interested in cancer are always interested in the translational effects, but you really got to know the mechanism of the biology to really understand like how it's going to be a therapy or understand more of the disease.
Right, and so I think today you see a lot of observational studies, and everybody's interested and excited about single cell and spatial and all this, but at some point, from a therapeutic point of view, which is still my primary interest, if you don't know exactly what something's doing, it's very hard to figure out how to make it therapeutic.
So yeah, we know you from that. So if you're a postdoc, a lot of the work was starting RB, and then afterwards you started your lab at the Dana-Farber, right? And I think what a lot of people know you for is like kind of ushering this area of cancer genomics. So like around in the 2000s, you, along with Matthew Myerson's lab, you pioneered the use of genomics. Specifically, that one example was to study these EGFR mutations in lung cancer. Could you tell us more how you and Matthew thought about this whole initiative?
Yeah, but maybe I'll say that's not how my lab started. Like, I don't think I would start a lab and say, oh, I want to sequence all the kinases. Like, that would seem like a really spectacularly bad idea. So when I was in Bill's lab, Bill was working on RB, and the day VHL was cloned, he's like, oh, that's a good idea. I'll work on VHL. Nobody knows how that works.
Yeah.
And so obviously he became very successful, won the Nobel Prize figuring out. And so I thought I was very interested in sort of helping the Farber build prostate cancer program. And the day my lab, or like right around the time my lab opened, P10 was cloned. And initially I was going to sort of work on trying to find like the prostate stem cell, like not the prostate cancer stem cell, but literally the prostate stem cell, thinking that that might be where the transformation events were happening. But the day P10 was cloned, I read the paper and I'm like, whoa, it's deleted in 2 out of 3 known prostate cancer cell lines. And by the next day, I was ordering PCR primers, making C and N terminal GST proteins, immunizing rabbits, and starting to work on how P10 functioned as a tumor suppressor. So that's how my lab got started. And then, we got an R1, we got a whole bunch of grants, we started publishing in that area. And then, because of our work on P10 AKT, I was sort of involved with Novartis and their funding mechanism. And Matthew and I were both involved in that, and sort of we reconnected. We had been connected before, but we reconnected and started talking about what's the implication for the approval of Gleevec and CML. And I think at the time, a lot of people thought, oh, this is kind of a one-off, right? That, oh, there's this weird disease, CML, it has this fusion. But we thought there must be other examples of that. And we had also been working with Todd Golub on expression profiling, and Todd was really sort of one of the earliest leaders in sort of the genome application of things like microarrays. So we had been connected with him through that this Director's Challenge grant. And we were just on a plane ride back, and we're like, well, there's got to be more of these kinases around. And we decided we'd get back, and he was interested in lung cancer. I was interested in prostate. We knew Charles Sawyers, who was interested in brain tumors. So we decided to sequence the tyrosine kinase domain in those around 100 samples from those 3 cancers.
Okay. Yeah.
At the same time, because of the microarray data, Matthew and I had both grown up in sort of the tumor suppressor area, and while sequencing was fine to find a mutation, loss of heterozygosity was still the hallmark of a tumor suppressor. And at the time, the sort of most dominant technology for looking at copy number was CGH, comparative genomic hybridization, which didn't tell you allele-specific copy number. So you couldn't really see loss of heterozygosity because loss of heterozygosity can either be haploinsufficiency of the allele or what's called uniparental disomy, where you duplicate one allele. So, for example, P53 often loses one allele and duplicates the other, so there's no copy number change. It just becomes monotonous in terms of variation through that region. And because of the connection with Todd and the Affymetrix connection, Affymetrix had the 1.2K SNP array. So we started using that, which was obviously very tiny, to look at copy number and LOH, and that led to the 10K array, then the 100K array, then the 250 and 500. So in parallel, these 2 efforts to sequence genes and then understand the copy number landscape sort of emerged out of those interactions really between myself, Matthew, and Todd.
It's really cool. Yeah, I think it's good, the benefit of being in the area, and I think it sounds like maybe the conversation started on this airplane ride, which I think is usually not the most productive. Usually it's not the most productive thing for people, so it's pretty amazing.
Well, we ended up by chance sitting next to each other. But, I'd say the other thing, just to go back to David for a second, Rick Klausner, who actually Bill had also interacted with on VHL, was the NCI director, and they started this thing called the Director's Challenge, and David was helping Rick sort of envision how to do more innovative science. And he came back and he called me and David, me, Todd, and Matthew into an office, said, we're going to write this Director's Challenge grant.
Oh. Wow.
And so we're like, okay, we're going to write this thing. And so the three of us working together was really David calling us into his office and telling us, like, yeah, go write this grant.
Oh, wow.
Yeah, so
Wow,
that was
fantastic.
one of the, yeah, he like I said, he had a big impact on my early career, and then even at this transition, he sort of fostered this collaboration that led to these different omics approaches to cancer.
That's a really cool story. I feel like now we take it for granted how this field of cancer genomics started, and it's all just from people coming together and being like, we're going to do this.
Yeah, it's funny because I think sometimes people think, oh, the Harvard environment is so competitive, right? Yeah, well, if you want to compete with Harvard, even if you're not at Harvard, you're competing with people at Harvard. Like, that's a given. But actually, most people at Harvard are, like, super excited to share all their ideas and to talk. And I just benefited tremendously. Like, none of this would have happened without, Todd sort of being in Eric's landscape and getting into the genomics, and Matthew and I sort of reinforcing each other's desire to do this. Like, none of it would have happened without the colleagues around us. You could never have done any of this sort of off in isolation.
Yeah.
Collaboration. Yeah.
And it's like an environment that promotes people sharing like info and presenting to each other like what we have now, like a cancer program.
Then you took the role of the VP and the global head of oncology at the Novartis Institute. How did your opportunity arise?
Yeah, well, basically I got a call out of the blue one day. Yeah, I just got called by Mark Fishman, who had made the leap himself from MGH and head of cardiology there to run and really establish this institute in Boston. They were looking for a new head of oncology. So I got a call, and at the time, we had just made the EGFR breakthrough, and things were going really well. But you could also see that there was this moment where it didn't take much to see, like, we're now in a position we will know all the genetics of cancer. We might not know them right at this second, but like 10 years from now, we will know the genetic mutations that drive cancer. You didn't have to see TCGA happen to know that that was going to happen. And still you'd be left with this question of, like, okay, if you have all this data, who's going to make the drugs in that specific, going after those specific patient populations or specific genetic alterations. And at the time, I would say there was still a lot of pushback on using EGFR mutations in EGFR mutant lung cancer. You'd say, like, oh, that's like totally obvious, right? You cannot believe how non-obvious it was to some people that, didn't want to accept the notion that we would profile patients, find their mutations, and not just treat them as one uniform lump of cancer patients. So the idea that you would have the potential opportunity to lead drug discovery in that venue was really attractive. The second was it was in Cambridge, so that really helped. I didn't have to move, and my kids were like in junior high school and high school, so they didn't have to uproot or leave. I have to say there's a lot of advantage not to having to make a big decision about your life and moving and uprooting everybody at the same time that you take a new job like that. The third was I would hopefully get to interact with the environment I was still in, so presumably all my friends would still help me, I hoped. The opportunity to, like, someone like Mark would say, this is your responsibility now, and it's kind of on you to lead it. That was both, like, terrifying at the same time that the opportunity to say, like, can I really do this? And the challenge of it was a lot. Actually, a lot of the draw was just like, can I actually do this? Yeah. So that's how that happened.
Yeah, I mean, it sounds like an exciting opportunity to be more involved in like developing the medicines for patients. And I imagine as an academic, I think NIBR now has a reputation where it's like industry, but it's still somewhat like academic with the discovery. So I imagine that was something that was enticing as well.
Yeah, probably even more so. When when I went, we sort of were in a good period of time from a funding perspective. Gleevec was sort of not even at its peak in sales. The oncology commercial business was doing really well, so there was a lot of investment. And so the Cancer Cell Line Encyclopedia, like, that came out of this idea that, okay, TCGA is going to sequence all these tumors. Well, what does that matter if you have no model systems in which to model the tumors? And actually, the Cell Line Encyclopedia was something in August of the year I took the job. I kind of knew I was going to take the job. I woke up one morning while I was on vacation and wrote a 32-page, like, here's what I will do in industry. And the Cell Line Encyclopedia was one of those things where, like, we need something like this, otherwise we won't be able to make the drugs we're talking about. And then, like, we had the funding to do it and to work with the Broad to bring that to reality.
Okay, cool. And I imagine you had the connections with the Broad because it was you at Novartis and it was Levi at the Broad?
So Levi was my former postdoc. He inherited my former lab. So,
oh, wow.
Yeah. So Levi was a postdoc in the lab when I left, and fortunate for me and for the lab, I didn't have to shut my lab down. He was reaching a point. He had discovered the MIT amplifications using those SNP arrays again in cell lines of melanoma. So this was like one of the first examples of a lineage-driven oncogene. He was clearly on the path to faculty, and so with my departure, the grants could turn over to him. And the first year I spent coming back and forth helping him supervise the lab and the lab meetings, and then he was on his own taking over the lab. But then we started this the Cell Line Encyclopedia collaboration with Levi.
It's really cool. So it was a pretty seamless transition, and you guys liked working together, so it's good you got to continue that at NIBR.
Yeah, and it was a different type of industry collaboration. I think in earlier industry collaborations, often it's like give money to an academic, and then they go off and do something. This was actually a joint project, and it didn't have like some sort of nebulous goal. It was like buy and grow 1,000 cell lines, extract DNA and RNA, sequence the RNA. Actually, we couldn't sequence RNA at that point very easily, but it had this large-scale collaborative project orientation towards it, which was quite different.
It's really cool. Yeah, it's definitely very ambitious, and I think one of the notable discoveries that was found from this was that there was this specific dependency with synthetic lethality of PRMT5, this protein arginine methyltransferase.
That came a little later, I would say. So once the Cell Line Encyclopedia was established, at that time, shRNA hadn't been discovered yet, and CRISPR hadn't been discovered yet. So this was in advance of that, but we knew we could use that to profile small molecules. But you're talking about a period of time where you could hypothesize a gene or protein was of importance therapeutically, but you had no way to test that short of trying to do oligonucleotide, antisense, knockdown, dominant negatives. So the advent of shRNA or siRNA clearly was going to change that, and some of the early data sets were the shRNA compared to CRISPR is quite noisy, and we decided to embark on this Project Drive, which would be a much larger scale effort to validate or devalidate genes by using, in this case, 20 shRNAs per gene, but not trying to do the whole genome. We did half the genome. And so that was an intrinsic project to Novartis, or to NIBR. So we had 3 target validation groups at that time, sort of reading the literature, looking for targets, most of the time devalidating targets, and we just decided enough of that. We'd take all 3 of those groups, and all 3 of them would work together to just do this one project, and then we would know whether all these things were valid or not valid all at once. And so that's how PRMT5 was discovered. It was one of the top hits. Werner was actually discovered, but we didn't publish it then. So that's why Novartis has the first Werner inhibitor in the clinic.
Interesting. Yeah. So this is the target is pre-selected. I mean, it's not whole genome. Sounds like.
Well, semi-pre-selected. You had to select roughly, I think it was around 8,700 genes. So I am sure we covered mostly enzymatic and druggable genes, but it wasn't so biased that you're just picking the kinases, for example. But I'm sure we covered every kinase there was, right? Yeah.
Oh, wow.
Yeah.
Yeah, it's really impressive. And I think at your time at NIBR, like, you guys were developing your classic small molecule therapies, antibodies, but I think one of the really interesting parts was this development of Chimeria.
Kymriah, yeah.
Kymriah.
Or at least that's the way I say it.
Yeah, it's easier to say that than tisgenlecleucel or whatever.
I can't say that.
Yeah.
We called it CART 19.
Oh, yeah. But yeah, you guys have developed this CAR T cell treatment.
Well, so not us. What that's the sort of example of like making sure you're in touch with the external world, right? So the catalytic moment there was Carl June's publication of CART 19 in 3 patients with, I think it was 2 with pediatric leukemia and 1 with chronic lymphocytic leukemia, if I'm remembering right. Or no, it's probably more than 3. But there were 3 patients who looked like they might have been cured by a single infusion of his CD19 directed CAR T. So there was this New England Journal paper. We all saw it, and I give credit both to our side for saying like, hey, curative therapy is not very common. Actually, it's incredibly uncommon, especially single dose or single modality cures are incredibly uncommon. But I also give credit to our development side, because a lot of development organizations would have said, like, there's no way we're going to do this, right? But our development organization was also pretty interested. And so having both the research side and the development side together, interested in this modality, helped then drive this collaboration with Penn. And so instead of Penn starting their own company, which they could have done, Novartis helped them to build a CAR T center at Penn, but then we developed a joint project with Carl. So Carl and I oversaw the joint steering committee for the CART projects. We had different CART targets going into the pipeline. So yeah, like, this was by far not my invention or anything, but I helped manage the collaboration and work with Carl to keep that moving forward.
Yeah, it's a really cool example of like such a seamless collaboration with academia and industry, and it's very promising. I think we've had kind of shortcomings with standard modalities, and hopefully with immunotherapy there's a lot of promise.
Yeah, I mean, CAR Ts are still, I would say most people are like, see these waves of everybody's excited and everybody's not excited, and I think it's the kind of thing where there will be a series of progressions where both the manufacturing gets better, the ease of delivery gets better, but also the ability to target a broader selection of tumors through different ways of engaging with the targets will happen over time.
Okay. So now you made the transition back to academia. I think usually people said it's one way if people from academia to the industry. So could you tell us that why you come back to start your lab again at the Broad? How different was it to start your lab? I mean, now compared to the first time, the second time, right?
Yeah, definitely harder the second time. You'd think you'd know more, but actually I think you know less over time. But so, I had been there for 11 years, and I thought that was a great 11 years to have. And at the time, Mark Fishman was retiring, and so the whole leadership was going to change. I would say the commercial landscape of oncology was also changing, with IO sort of having this very big sort of panic effect on the world of drug discovery. So, it just made all of those events came together to make me think that, like, okay, I've done this from when I was like 45 to when I was 55. Novartis actually had a policy to allow you to retire at 55 if you had worked for 10 years. So there was also some sort of convenience to that. And so I just started thinking about, like, what would I want to do in the next phase? Because if at 55, if I went to 60, I was pretty sure starting a lab at 60 wouldn't be the best idea, or I didn't think that'd be a good idea, or making a transition might not be the greatest thing. But at 55, you still have quite a runway. So I just started thinking about, like, what do I really like to do and what do I want to do? And do I do this same job for the next 20 years, next 10 years or not, right? So, that was kind of the thinking. And then there were several options, like, do I want to be something like a cancer center director? That didn't seem like the kind of thing I would want to do after industry. I think you have to be much more sort of herding the cats more than telling the cats where to go. And then, or like start a small biotech company. But I still really love the lab. And I thought if I start a lab, and especially in the Cambridge environment, and I have ideas for biotechs, then I can engage and, both have sort of this industry footprint where I'm serving on an SAB or as a founder on a biotech company, but then, working in the areas in the lab that I'm still passionate about.
Cool.
Yeah. Was there like anything you kind of like, really missed, making that transition from academia to industry? Was there something where you're like, oh, damn, this is really nice?
I would think there were things to adjust to. So one is that in industry, the youngest scientists are the most experienced in academia. So your most experienced postdocs become the junior scientists in industry, and often the research associates are also already have 2 or 3 years of experience, and then they might move to industry. And I think you get used to the unbelievable experimental capabilities, not just sort of money or technology, that like people can execute experiments because they've just done it a lot. They're really good at it.
Yeah.
And so the fraction of highly successful experiments just seemed routinely extremely high. And then you sort of come back to academia and you realize, wow, there are people who are still learning a lot. And, and my expectation of how fast things would happen was off by years. Like there are things, I would say, projects that I thought we'd be done in 6 months that took 2 years.
I think industry is known for efficiency. It's like academia is not efficient.
But also, it's not just efficiency. It's also that there's a very different level of training, and so that it took a while to get used to that. Oh yeah, I have to be patient, and that people are still learning how to do all this stuff, right? Yeah. So I mean, that's also the exciting thing, is to see all the young people coming out of college who are like super excited about everything. That's like cool too. But getting used to this sort of the difference in the experimental pace. And I would say the pace in industry was also, like, I think people sometimes think it's like slow and lumbering. That was not my experience. It was like high paced. It felt like, being on adrenaline.
Yeah, I imagine. So there's pros and cons to both.
Yeah.
And then I guess one of the areas your lab was focused on when you restarted your lab at the Broad was, the return of PRMT5. And, it's known for its synthetic lethality in these MTEP-deleted tumors, which is commonly deleted because it's next to CDKN2AB. And you guys had an interesting approach where you're interested in its interaction with these substrate adapter proteins. So could you tell us more about this kind of initiative? And
yeah. Well, first I would say, like, you're coming back to a new lab. It's not like I'm going to work on P10 again, right?
Yeah.
And so what are the things that are influencing you to think about a new lab and what you do? It's all the science I was just doing at Novartis, right? So I don't want to hide the fact that, like, half the stuff I was thinking about is all the stuff that was current in my mind. So if we take PRMT5, so I was pretty convinced someone would make an MTA cooperative PRMT5 inhibitor. That's the mechanism that we had proposed would be synthetic lethal. And I think that has now been borne out by drugs from like Mirati and Tango and Amgen. Interestingly, Novartis did not make one. I'm not sure exactly why. But I also knew that at some point there would be resistance. Like this sort of the natural way things go. And we had this experience with BSAR-ABL of making 2 different inhibitors to the same enzyme, one that bound to the myristyl pocket and, of course, the catalytic inhibitors, and showing that by binding to this critical enzyme in 2 different places, that cancer, at least in this in vitro and in vivo model systems, really couldn't find an escape way around that. So this idea of dual inhibition of an enzyme that was critical to a cell was also in my mind. And so I came back to Novartis, and I didn't want to work on, like the new MTA cooperative inhibitors, but we also knew that in our screens, the genetic screens that had shown the synthetic lethality with PRMT5 had also shown that the substrate adapter proteins, Rio kinase 1 and WDR77, were also synthetic lethal, which almost certainly meant that the interaction between PRMT5 and these 2 substrate adapters was going to be critical. So I thought, like, okay, a second site inhibitor would be one that blocks the interaction of those two proteins with PRMT5. So that was the idea. And we did eventually discover the mechanism, actually through some great work by a crystallographer in CDOT, Brian McMillan, and then Kathleen Mulvaney, a postdoc. But yeah, interestingly, we had a lot of trouble actually validating the genetics of that interaction. I still think it's probably right, but we had trouble like proving that you could get deep anti-tumor responses by genetically manipulating that interface. And we did have a drug discovery project which led to a first covalent inhibitor of that site, but also we didn't have a lot of tractability on that particular chemical matter. So that's sort of those reasons led that project to a dead end over time.
Okay. This is pretty cool to hear, maybe like the use of like the PRMT5 MTA cooperative inhibitors as well as the PRMT5 substrate adapter inhibitors as well.
Yeah, I mean, right now what we're doing is studying still. We've done, for example, anchor screens with the new Mirati's inhibitors looking for cooperative combinations, and we have a paper under review that will show in a RAS mutant background, there's pretty strong synergy between an MTA cooperative inhibitor and either a RAS or BRAF-CRAF inhibitor in the double mutant population. So in pancreas cancer, a lot of cancers are mutant have a deleted MTAP and are mutant for KRAS. So the fact that there are two independent driver events also might lead to the creation of a combination of two independent inhibitors that then may or may not show cross resistance. And if they don't, then you would have a significant advantage over the monotherapy.
Oh, wow. Okay. That was a really good story. Thanks for sharing that.
Yeah. So another area you work on relates to the called context-specific gene activation induced lethality. I think that's very recent, right? You get the paper published. Yeah.
Yeah, it's recent for us, but it's sort of the history of that is pretty old, right?
Yeah, sure.
So it even goes back to people showing that if you just overexpress KRAS, cells undergo senescence instead of transformation. So in some ways, the too much of a good thing is not exactly a new idea. But we have found that you recurrently run into this. So we first actually just saw this in 2012 when Meghna Das, who was a postdoc at Novartis, was treating mice to resistance with vemurafenib, a BRAF inhibitor, with a BRAF tumor. And when she stopped the drug, the tumor started to regress off treatment, suggesting that there was some sort of addiction to the drug per se. And they went on to show what was happening is when you withdrew the drug, ERK overactivated, the tumor regressed transiently until ERK renormalized, and then once ERK was sort of back to the Goldilocks zone, the tumor started to grow again. But you could actually cycle on and off the inhibitor and keep the tumor at bay longer than just continuous inhibition. So cyclical therapy, at least in the mouse, was better. And then a second postdoc showed that if you just turned on ERK at the beginning, you could kill a BRAF mutant tumor. And then because of our interest in functional genomics and because we were interested in this idea that the current generation of single gene knockouts might be missing things like CDK4/6 and MEK1, MEK2, ERK1, ERK2, AKT1/2, BRAF, CRAF, there are all sorts of these paralog combinations that might be missed in single gene knockout screens. We had built a paralog library. This was work done by Takahiro Ito, and Tak made a paralog library and then screened NRAS mutant melanoma plus some others, but found one of the top paralog pairs in this case was DUS4/6. So you knock out DUS4/6 and NRAS mutant tumors die, and it turned out DUS4/6 is a negative regulator of the pathway. So we knock out DUS4/6, ERK goes up, and NRAS mutant cells. Yeah, so we then, we followed this up by trying to do straight gain-of-function screens in the PRISM assay in 500 cell lines, led by Liang Chang, a graduate student. And he found more than one pathway can be susceptible to this in a very specific manner.
That's really cool. And then you mentioned Liang. Liang is a former member of our
club. Former member of Biotech Club.
Fantastic.
Yeah. And then that sounds really great. So this concept's been around for a while, but I think people can't imagine how it's used therapeutically. And we know you started this startup, Delphia Therapeutics, which is looking to create therapeutics based on this concept. So I think that's the thing. It's like, how do you develop these therapeutics to activate or promote expression of these genes?
The easiest way to think about it is we're actually doing this more than we think, right? So anti-PD-1 therapy is blocking an inhibitor of T-cell activation. So if I told you I'm going to make a T-cell activator, maybe you'd be afraid of that. But if I tell you I'm making a PD-1 inhibitor, no one seems to have a problem with that.
Yeah.
Right? Yeah, so if you think about activation as inhibiting the inhibitors, then it's not that different than things we've done. So if I called it a DUS4/6 inhibitor instead of an ERK activator, yeah, maybe people would have sort of a less of a reaction. That being said, I do think, yeah, there are hurdles. The most common one people think about is, oh, aren't you going to cause cancer? I think this is the least of the concerns here. Cancer is generally a multigenetic process. It doesn't just happen by turning pathways on and off. It happens through genotoxic events that cause mutation. And we've dealt with pro-cancer effects of chemotherapy. Etoposide in breast cancer patients does cause longer-term risk of secondary malignancies, but that's because etoposide itself is damaging DNA. So I don't think an activator, unless you make a bad one, is going to be damaging DNA. So the risk on the pro-cancer side would be more that you have a pre-existing lesion that has like a RAS mutation, and now you turn it on, and maybe that pre-existing lesion becomes worse. I would say, alternatively, the pre-existing lesion could also undergo senescence, for all I know. So I'm less concerned about that, mainly because at the time we're treating patients, they already have a lethal metastatic disease that needs to be cured. So the first step we have to do is cure the disease. I think the more complicated aspects are sort of dose and schedule. It's very unlikely these are going to be continuous. You can imagine the dose has to be at a certain dose and a certain level of activation to get inhibition, but starting at a low dose might not be the best way to do that. So I think talking to regulators about how to start trials closer to a therapeutic dose is going to be important. And then I think there probably will be unexpected adverse events, just like there are with most therapies, and so you would have to deal with whatever those happen to be for the mechanism you're working on.
That's a good point. It's like a new area, but there's always issues with others. And it's like you have to kind of you have to confront them, and then you'll figure out like how to address the obstacles.
Yep.
And then so we kind of talked a little bit about the research as starting your lab, and one thing that like we know you're pretty big on is this like insights. Personality training that you encourage your lab to do, and you talk about. So, can you tell us a little bit more? Because we know your personality type's red, right?
Yeah, bright red, pretty much.
Red.
Yeah. Well, I think I will say in my first lab, and when I was sort of a postdoc, I felt like, oh, I'm a postdoc, and to me it was like you make it or you don't. You just sort of felt like it was kind of like up to you. And that just seemed to be the way it was. And then when I had my own lab early on, I also thought like, oh, people just need to, I'm giving them this opportunity. They need to make it or break it, right? It's like up to them. And also I had very little, if there were interpersonal issues in the lab or personality conflicts or things like that, I had no sort of tools or training on how to deal with that. Or even feel like I should be dealing with that and that it was my responsibility. Yet I sort of knew that there can be personalities in the lab that are making everybody unhappy, right? Or a lot of people unhappy. And I think I learned much more about that in industry where they, like you don't have people coming and going. You have people coming and you want them to stay. So you want to create this culture of respect and cooperation and collaboration. And industry does a lot to try to, improve people, mentor people, train people. And so I think I just got, either because I got older or because I had more exposure to it in industry, I got more training in that. And insight seems like a really interesting way to do it because it focuses not just on telling you like you're red like I am or yellow or blue, but also different communication styles and how you could use the fact that I'm red and I overtalk people. How do I modulate that? Or if you're dealing with me, don't send me like a 3-paragraph email because I'm not going to read it, right? Like you have to know who your audience is, and I think insights is really interesting in that. But I will also say, just doing it, even if let's say the impact is 10% of people improve or get better, the fact that people know you care enough to do it actually changes the attitudes in the lab. So that's the most interesting thing to me, that if the lab knows you care about their behavior and culture, actually behavior changes it for the better anyway. So people know that I'm not going to tolerate bad collaboration and cutthroat behavior because we've talked about it. And that alone, I think, makes a big difference. It's not perfect, but just sort of saying you care about that, I think, is a big step forward in making the lab environment better.
Yeah, it gets a conversation started. And then, I know a little bit of this just from conversations, but the Insight gives you a specific role or like personality. So your role is pretty
director.
Yeah,
it's weird that it's actually a little scary that you can take like whatever a 40-question questionnaire and it spits out this like, here's what you're good at, here's what you need to work on, and like you read that and you're like, yep, everything's like, it seems like we do fall into some of these personality types, and the Insight seems to be able to pick up who you are pretty quickly.
Okay, I think we can wrap up. The last question is, so what's are you hobbies outside of work?
Well, I would say I'm trying to cycle. I'm having a lot of trouble having time to get on the bike. So cycling, I would say, is like trying to stay in shape, and mainly through cycling is the main one. And then we've been with my wife and sometimes with the kids. We travel. So travel, cycling. I love cooking, but I haven't had a lot of time to do that.
Still a lot of coding.
No, zero. I actually did. Okay, during COVID, some Christmas break, I took an online, not Python. What's the other common?
R.
R. Yeah,
R.
I took R.
R,
I took
yeah.
R. Like I learned how to make graphs and stuff and program in R and load data. But like like if you're not using it continuously, it's really hard. And I have to say, what was
Rusty.
Yeah, you get rusty fast. But what was annoying to me is still things like parentheses and periods and stuff like that. Like I would have thought by now, okay, that was like when I was coding in Fortran, if you missed a period or had the wrong parentheses, it would mess up. You would think by now it would be like all auto-correcting all that. And I was very disappointed that was not the case.
Yeah, it's so particular.
Maybe ChatGPT will fix all that.
Cool. And then you mentioned a little bit about cycling, and I see online you participate in this Pan Mass Challenge with Dana-Farber a lot. So imagine it's like you're very big into cycling if you're able to do that, because that's the ride across Massachusetts.
It starts. I start in Wellesley, so it's about 170 miles, and 100 miles for me the first day, and then 70 the second day.
Oh wow!
But honestly, like that's not that hard for me at any rate, because it's pretty flat comparatively. I've done some more cycling, like in Italy and with some friends of mine, and they are way beyond my category, and all they like doing is finding the biggest mountain they can go up, and then you're going up forever. So the Pan Mass is like not that hard, actually.
Yeah, it sounds like a breeze compared to going up a mountain. Well, thanks for sharing more about that, and we'd like to end this conversation by saying thanks for taking the time.
Yeah, thank you for your time.
I think this podcast is the pulse because we wanted to talk to people that have their finger on the pulse in terms of what's latest in biotech, and we thought you'd be the perfect guest because of your experiences in academia and then industry, and still have all those connections, in academia to the industry people as well. So it's great to hear all your insights.
Yeah, fantastic. Thanks so much for doing this.
Yeah, well,
Thank you so much.
Hi everyone, that's the end of this episode for the Harvard Griffin GSAS Biotech Club's The Pulse podcast. I want to thank Fung for helping co-host and produce this episode, and I want to thank everyone for listening, and please stay tuned for our next episode.